Okay, so I'm going to start to get on. Thank you very much coming together. My name is Dr. Robert Lyons, and I'm going to host this today, this function about the immune cells function hypoxia and how to relate it to COVID-19. And I will get into a presentation soon. I would like to have stated to you there's two different ways to look at this disease, because from the day one is very controversial. We don't know what is this virus. It's a new virus. We don't know what it does. You know, actually the numbers are not matching up right. We don't know if it's real or not. So in the end, I don't want to end up saying to you something what is not right. I tried to put together my presentation last night, and all the numbers was valid last night. It was not valid this morning. So I'm not trying to get into if it's right or not, but we know one thing: how the immune cells should work, and what we can do to develop a defense mechanism against virus infection. So I would like to get into my presentation, and I would like to get into the first slide. You might notice that if you look at the mainstream media, they tell us that was the number yesterday. It was 4,311,914 people being infected. This is kind of very accurate number. And, I'm looking at the death toll is 290,597. And the recovers is 1,567,141. And the currently patients who's trying to get through this disease, some of them not even going to feel it or not even going to notice it went through. Some of them going to be extremely ill from it and maybe end up dying. This is a very interesting numbers, and that's what the whole world looking at and try to follow this. It is actually from WHO recommendation, and all this United Kingdom numbers, it's death toll is 32,692, and a lot of cases 226,463 cases been infected, and the numbers are just bouncing there for the last couple of weeks, and now they're trying to open it, and they think they are over the curve. And let's go a little bit more into this, because I'm sure you know this from the media. Every person affected different way. If you look at the syndromes of fever, they told us in the beginning you might get fever, you might get dry cough, tiredness. We can get this anyway. We don't need to get a COVID virus. We can have any virus, and even just a small bacterial infection would do the same. The more common syndromes is aches and pains and sore throat. Some people get diarrhea, conjunctivitis, in the eyes, get infection, headaches, and the most significant marker is the loss of taste and smell. So, that's, and discoloring the toes. I would say most of the viral infection comes like this way. So this is just like somebody put together a Rubik's cubes and, put everything together, and one virus can cause all these syndromes. When it gets serious, again, I say that's what they told us. When it gets serious, then the difficulty of breathing, shortness of breath, chest pain, pressure on the chest, like somebody sitting on the chest wall, loss of speech or even movements. You cannot get up. So, all these people who has this, they should really try to get into a doctor office and contact your doctor and see if you have this virus or not, actually. All this about, one more slide we have, it takes about, the average infection times is 5 to 6 days, and and the virus should have some kind of a syndromes. They say it could be 14 days even to go, but I heard some other news media 21 days. could happen. So all this not really clear, but we know about virus for many, many thousands of years because virus is living with us for a long time. And we know this, all this effect. So humans are right now in great danger. That's what they're telling us because we have to stay home. We cannot go to work. We cannot do anything because of this virus, which is mean we don't know what is it and where it's going to come from. And so there is a lot of question what is not answered on that. So, but let me tell you, on my part, what I'm thinking is it's worth to listen and learn to follow. So let's see this. I had some search on this. There was a test on COVID-19 2020 in Australia. That test was very interesting. It was Melbourne, Australia. They tried to find some kind of a case reports on COVID-19, and they were testing cellular immune system functions. And they find out all the T cells and the B cells activation showed a remarkable answer on this study. That means if a patient has some kind of a reaction after 2-3 days of the virus entering into the cell, after that you have a very good possibility to respond on it. If not, that means you are very tired. You run down your system, you have some long-term disease, you have some viral infections or some fungal infections, some bacterial infections, but break it down. What they come up with, they said we have to be aware of it when COVID-19 attacks the T cells and the activated CD4 cells, which is mean cell-differentiated co-receptors of T cells and CD8 T cells co-receptors. Together, they are really much seeable by binding the antibodies in the patient blood. So this is very important when you have the symptoms and you have availability to look at your cellular immune system. The first thing is becoming to the T cells will be activated. It must be because the T cells and the CD cells, the CD8 cells, they are all trying to respond. The same thing happens with the AIDS virus. With AIDS, HIV virus, when you have AIDS, you have, the same issue in the white blood cells, the T cells, the CD4, CD8 T cell co-receptors being watched. If they are too low, they are not capable to fighting the virus. And that's where all but it takes 5 years, 10 years to draw your immune system down with HIV positive. And with this virus, it looks like going very fast in some case. Not all the time, but some case. If somebody has low immune system, this is the first defense mechanism. The CD4 and the CD8 cells and the natural killer cells will drop down. So let's see. In 2018, there was a Nobel Prize winning two professors, one from Tokyo, from Japan, and one from United States. They stated that on cancer study, also if you have the T cells activated, if you have T cells activated and the B cells activated and the natural killer cells activated, you have some kind of a good results to fight cancerous cells. As I said, this T cells is findable in the white blood cells, and it's, today with not a routine, but with a laboratory test, you can really go after those guys and see it. They stated it, if they have a chance to somehow elevate the T cells and the natural killer cell functions, they could have developed new drugs and new methods to fight cancer, because this is much better and much more longer lasting, better effect just to use the chemotherapy or radiation therapy, or even you can use it together. You can use it together with radiation therapy or the chemotherapy. So I think they stated that in the future there will be a lot of studies going into these T cells. And then what I wanted to tell you, in 2009, we've been working with the Kaqun water since 2002, December 27. And by 2009, we managed to get to the first study what came for human trial study with ethical medical license. Actually, it was 2009. It was the National Institute of Chemical Safety and Cytogenetic Immunology Department. And this study went on Kaqun water, on the immune parameters on healthy volunteers with ethical medical license. The most important part of this study was actually designed to see how long we have to drink the water, how long we have to take the baths in this water, and what kind of a immune response we're gonna get if somebody using this water for 14 days. And keep it in mind that was 2009, and everything what I told you earlier was 2020 and 2018. So we did publish this about 9 years earlier than anybody else. To one of the most important defense mechanism is to elevate natural killer cells, their function, T cells, B cells, CD4 cells, CD3 cells, CD8 cells, CD25 cells. So all these parameters was watched very closely in this study because we wanted to know what kind of the response we will get from the patient. That's all started it where we had 20 different patients with 20 different disease, 20 different age, and all of them responded right with the therapy. We call this immune boosting therapy. Everybody responded right. Why is that? It's because, I never heard of any drugs like that, what is good for this, then good for this, and good for another. And namely, if somebody has cancer, maybe the medication is different than somebody has ulcus, or somebody has a diabetes ulcer, or somebody has systematic mycosis disease, so fungal infection. Everybody is all this is all different way they grow, different they spread in the body, a different way to feed that or kill it. But somehow we needed to find out what is going on, because every patient who entered to the therapy in this test, we saw the improvement. So we were in 2009, we were very, very happy to find out what activating in the cellular cell, but we did not know why it's all of these cases is good. So we had to study this over and over to find out. Okay, and then the next is we try to move it. So it is really was revolutionary. Think of a simple water with highly oxygenated water, which is not bubbling, there is no gas. And this water is capable to elevate your oxygen parcel pressure by 15 to 20% every time you drink 3 deciliter of it. If you take a bath in it, it could be much faster and much more efficiently. So the study went with 30 patients. There were 15 women and 15 men. And the study was conducted from 0 point all the way to 21 days. 0 point means the 0 point they took blood from the patient, and after 21 days they took a blood again, and between that they took every 7 days one. So there was four blood tests they were taken, and we find out without exception every single patient who entered the study was somehow activation of the T lymphocytes. And that time it was a major breakthrough, because you cannot just elevate this. There's no medication on that. And that give us proof the oxygen what entered the cell, entered the mitochondria, and restarting mitochondrial function and stopping hypoxia to elevate the activation of the T lymphocytes really proved that to us. It is really taking place after drinking the water and taking the bath for 14 days. The percentage of natural killer cell showed statistically significant increase also. And what I tell you, because I was watching closely that study, the women are responded much better on the therapy. That's also very interesting. Women are responding much better on the oxygenation therapy in the water than male. We did not know that time, in 2009, why is that, but that was the fact. And then they watched the percentage of reacting oxygen species, to reducing cells increased significantly, so there was not much of the pro-oxidants because the body has the ability to fight off extra oxygen, so you cannot over oxygenate your cells. Also, we detect apoptosis. We also see the apoptosis was started up through Fenton reaction. And we also checked CD25 cell surface antigens. It showed us there was a real cellular immune system function. But it was really also be notable to know the antioxidant capacity increased significantly. This is actually great because we know that by now, if somebody wants to have effect on the HIF alpha, 1 alpha, you need to have antioxidant capacity grow. And actually the substance is vitamin C. You know vitamin C. Vitamin C is a very good antioxidant, but if your body is not capable to expand that capacity, no matter how much vitamin C you take, it will be not successful. But if we make the availability for antioxidants to grow and we make space for it, actually we saw the study, it was 72% of the samples, the blood samples we sent in, it was very, very positive. It was significant growth on that. And also we made the immunology division, I said, and then this is all the study you see. Mostly the final, what we need to talk about this and why is close to COVID-19, because COVID-19 is attacking the same immune cells as I showed you earlier as we studied, and this is the two what is very, very important to know. Number one, there will be no any loss of blood or quality or quantity of the blood being detected in the 21 days of the therapy. The second one is the natural killer cells numbers was growing. It was much bigger than from the 0 point, and they became a functional impact. Why is that important? Because more natural killer cells and availability, you have more availability to kill viral infected cells and tumoral cells. It's not mean this is curing cancer alone or curing anything. It means you have more availability to fight off viral infected cells and cancerous cells. Also the T lymphocytes, as I said, the CD25 cells were showing the antigen, surface antigen to elevate, and we were very happy about that because we could prove that we have cellular immune system activity. That was a response on it. So all these studies and all these literatures came out, and we published this 2010 at the oncology medical paper in Hungary, and the GPs also get this own medical journal, and we did put this into their journal too, so they could learn about it. But time goes by, and time goes by, and everybody forget about this. So our job right now to get more proof on that, we are not alone to just running around and listen to the media all the time. We have to see science. Science is very important, and you need to know the history of how humans evolved. And I can tell you, without natural killer cells, there would be no way for us to be here today. Because I tell you, and it's not a secret, There is many virus infections was before, the back plague, the, all the malaria, all the viral infections, herpes simplex, all the HIV, all the infections, SARS, all those are, if the human body can not fight it off, nothing will. That is something we can do. You can have, this is many people have different opinion, and I still don't want to sit there and decide which one is right, which one is not, because they say we have to wait until the vaccination comes, we have to wait until medication comes. I believe that. I believe that either way, one of them, but to winning this war against COVID-19, it will be our own immune system. If our own immune system is working, functional, then we are gonna able to pass this, with the least suffering. We can go back to work, we can enjoy life, and we just have to test it to see it. Just an advice for you guys, but we always keep a good distance from each other. If you go into very crowded places, put some mask on. Soon as you get out, take it off so you have some fresh air, and all those a very important issue. If you have to cough, cover your face, and if you have a chance, get this water, look into this. If you need, we send you some studies to look at it, and try to bring up the immune system in a way where you could be protected anywhere. Important is eat right, nourish yourself with liquid. The liquid intake is very important. On any viral infection, you lose a lot of liquid. So please nourish yourself, not just with Kaqun water, drink other water too, drink teas, eat soup. Lots of liquid needs to go, because number one, not just bringing the oxygen to the cell is important, but the water is helping you to cool down your system. If you would overheat, or the virus trying to overheat, everything goes starting to invade you, the first thing is going to do, they dry you out. So nourish yourself. That's why it's so important to have the liquid replaced. And let's go for the next. Good news was 2019. It was already oxygen was in topic again, and that's why they get 2 American and 1 English professor get a Nobel Prize on this, because they said we have availability to accept oxygen to ourselves. We are able to sense and adapt to oxygen availability. That is a very important discovery, because before that they thought is only way to bring oxygen to the cells if you breathe in oxygen through the lung. Now we know there is many ways to bring oxygen, not just through the respiratory system, but we can bring it into liquid too. And that liquid brings you availability to your body liquids. So if you mix your body liquid with this liquid, it definitely gonna be one thing different. The different will be your liquid most likely not gonna have that much high oxygen parts of pressure than the water is here, because that water has much higher oxygen parts of pressure. And between that two, it's very easy. They just have to balance it out. But again, the body capable to recognize if there is oxygen and capable to control it. That means you cannot over-oxygenate yourself that easy, because the cells has their own defending mechanism about that. So please look into this in this website. Please look at it, because that was also very, very important. Another advice, to take herbs, take exercise, take every measure you can, but if you must have normal oxygen parts of pressure in your tissue. And that's what we're offering to you. And I'm sending this water all over the world, and I'm sending this water to the hospitals now, to doctors and nurses, and I'm sending to all the way to the police department, all the way to the border guards, the people who are dealing with everyday danger, if there is a danger, make sure they have enough defense mechanism built in 14 days. And let's go. This was 2019, but 2010, we did the same study. Somehow we were a little bit back page on all this, and we had this study coming out. Actually, it was an in vitro study, not a human trial study, but it showed exactly the same what we are capable to do it. So before I close my presentation, would like to show you what I mean. If I had 8 different patients and put them in this water, and this water filled up the tank, 260 liters of Kaqun water, and they immerse in it for 50 minutes, then we saw 8 different patients, as I show you, and it comes up a little bit slowly, but here is the diseases. This is all different type of disease because we use the same therapy, different kind of illness, different disease, different sickness, different person, and everything is the same. We put them in the water. The question was, what is the common factor on these cases? Why and how we gonna see this? Why we seeing all this? Let me show you. The answer was hypoxia. It must be reduced to normoxia and activated cellular immune function. And if you look at that, after 1 month of period of time, each of the people who was on it, they all have recovered. Again, they had different disease, different person, different age, different gender, and the results is the same. So we had to find it, and that's what we have seen it, and that's why we did all these studies, because we wanted to know the truth. And I tell you, the only way they can be helped, these people, if they have the hypoxia stop, first place, oxygen deficiency of the cell, or lack of oxygen to the cell, and the cellular immune system function, namely the natural killer cells, the B cells, the T cells, the activated T cells, cytotoxic index. All those need to be watched in order to go forward. Okay, so I hope it was informative for you to see, and I would like to give my good friend the words, Adam Boday, to talk. Thank you so much for listening.
Thank you very much. It is very difficult to speak after such a good presentation of Dr. Robert Lyons. As a researcher, I would like to go a bit deeper, because we are very interested in what is underlying behind all these different diseases and how these different diseases are getting better with the same treatment. And the answer is in the title of this presentation, is the immune cell function. But in order to understand the immunity, the natural way should be studied first. And the natural way is always around us, and we don't have to do randomized, placebo-controlled trials in order to gather the information. We have to emerge into the biology and the ecology of the natural systems. And I would like to go into these backgrounds, because somehow we figured out that when we use the same treatment and we can treat or make therapies for different diseases, and we have good outcomes, then some kind of natural law is rediscovered in this way, like the discovery of the hypoxia treatment.
Yes.
If you treat the hypoxia and you ensure the oxygenation of the tissues, then all the conditions are getting better. Yes. In this way, it is somehow further away. And what is in behind us? Please, next slide. The natural immunity is working when the humans are in natural environment. The human biology is mainly constant for more than 200,000 years from now. So the people 200,000 years ago shared the same biology as we do now. And they were in close connection, surrounded by the triggers of the ecosystem they were living in. It means that they were adapted to the climatic conditions of their habitat, and they were every day they were in contact with nature. And what was in nature? Who was here first? That is the first question. If you go to the tree of life, 3.5 billion years ago, the life started on Earth with the bacteria. And the bacteria are doubling their numbers or propagating in, let's say a rough number, once in an hour. So it means that they have 10,000 generations in a year. And they are here for 3.5 million years ago. Billion years ago. So they are the ruling life form of Earth. And we are a complex life form, but we somehow in the last 100 or 1,000 years, we detached from this natural environment. We became so far from our ecosystem that we experience different biologic signs. And I am purposefully saying signs in this way because since we experience some kind of malady or disease or discomfort, it is a sign for us that we should change our behavior, which led us to this discomfort or disease. But in this modern life, we don't know what went wrong. How did we lose the oxygen from our tissues? How did it happen? And I would like to enhance the role of 3 organs, which we didn't really emphasize lastly: the tonsils, the Peyer's patches, and the appendix. Mm-hmm. They were told that they are just there, and it is just God's mistake. And some scientists a century ago said that the tonsils are and the appendix are useless for the human body.
They can just take it out.
Yeah, they can be taken out, and they only cause problems. If you go to space, they take out the tonsils, they remove your appendix. If you go to the space station, then you shouldn't have any problem there, because they cannot help you there. But what is very important, the tonsils are the gatekeepers of the oral cavities, and everything enters into our body through the noses, the ears, the eyes, and the mouth. The air, the water, and the food. Everything comes inside us. And the tonsils are surrounding the gate, and they are taking samples from the air, from the food, from the salvia we are producing, and from everything, from the dust we are breathing in. And from this air, they have small bays. And in this small bays, they incorporate bacteria and fungi and spora and everything, and they analyze it. And in the tonsils, it is full of lymphs, so the immune system starts here. And these information are processed in the whole body in order to have a specific immune response. Specific means that we don't need to contact loads of microbes. If we contact a load of microbes, then it is a danger that our body cannot stand up against them. But if we contact with small amount, with big dilutions, I mean 1 or 2 pathogen bacteria enter into our system, then our system can understand what are they, and they are preparing the T cells. From the tonsils, the information are first enters into the body, and the information is translated in the immune system to start the immunity against those particular ones, because the variety of the bacterial life forms is about, it's an estimation. It is always, you know, like the COVID, not really measurable, but a rough estimation says that it is about 30 million microbes surrounding us on the size of the bacterial scale. If you go beyond, and that's what interesting in the COVID problem, that it is estimated that 300 million viruses are surrounding us. But it is just an estimation, since we don't have a reliable way to measure it. Yeah. So the pair patches are the next phase, because through the tonsils everybody got into the system. But what happens in the system? The digestive system has small nodes, and these nodes, lymphic nodes, are full of sentinels, and they live in the pair patches, and they are working the same way as the tonsils. What is very important, that the fermentation starts. We have bacteria. I guess you all know this, that we have bacteria in the oral cavity, we have in the stomach, we have in the small intestine, in different community and different concentration, but we do have them. But in order to check how the fermentation and the digestion is working, then in the pair patches, we are constantly, vigilantly looking the bacterial composition of the lumen, of the material inside the digestive system. Why it is important? This is very important because these are the informations that we get in tuning how the digestion work. From the pair patches, we do the survey what kind of population lives inside us, and if we can, by normal, natural ways, then we do what we can in order to elongate the transit of the food or fasten it, like constipation and diarrhea, the two extremes of this. If we have very good material and very beneficial bacteria, then we experience the normal operation. If we are going to one extreme, then we have constipation, or we have diarrhea on the other end. This is how the gut is making a non-specific immunity against the composition of the bacteria or the invaders. And now the appendix. The appendix is important because the appendix is like a safekeeping device for the microbiota, the beneficial bacteria in the gut system. It is so small, and this is just a dead-end tube. And this dead-end tube is very useful because sometimes we would like to get rid of the bacteria because the pair patches say that they are not really helpful for us. We are experiencing a very severe diarrhea, and we excrete everything out, even the good ones, even the good bacteria. And then we need something. It's like a chamber, a treasure, where we keep our beneficial bacterial partners, and they are living in the appendix. And the appendix pumps it back to the colon, and in order to do the fermentation in a natural way. So it is also very important. But the appendix should be working properly. The appendix can be overpopulated by pathogenic bacteria, and that is the appenditis, when an inflammation starts there. But what triggers this? The only thing that triggers this is the dysbiosis, which occurs in the appendix, in the treasure, when we don't have order in our chamber of safekeeping treasure of bacteria. And that's why we have to put the question, why do we need vaccination? Because this modern society, the humanity right now, is in a health condition that one way or another we are relying on external artificial vaccination. Why is it? It's very important to know. This is the reason because we don't meet in small concentration since we were born. Can I have the next slide, please? We don't have the contact with different viruses, with different bacteria, with different fungi, with different microbes. And I would like to show in this diagram something very important. With the microbes, in order to decide whether you should do the vaccination or no, it's a statistical analysis. On the horizontal axis, we see the contagiousness of a disease. And on the vertical axis, you see the deadliness, the mortality. It means that on the horizontal axis, you see that from 100 patients, how much can 100 patient who has a close encounter with someone who is infected, how much will be infected. And on the vertical scale, you see that from the infected ones, how much will be dead causally from that disease. And that is also the causality from COVID is not certain right now, so we cannot say anything. These numbers are estimations from the recent scientific publications. But as you can see, the right-hand side is the side when you have to vaccinate, like the whooping cough or mumps or malaria or measles, which are highly contagious diseases. If the disease is not so contagious, it is less than 3%, then there is no need for vaccination in this case. The other way to, the other precaution is to do the vaccination is when the mortality rate is very high. If the mortality is very high, then we have to take it into consideration whether to do a population-wide vaccination, because we don't want to encounter this kind of bacteria in order to have the natural immunity, since 12 people out of 100 can be the casualty in this way. So this is a reason why we need the vaccination.
But if we don't get to that level,
if you don't get to that level, then there is no need to think about the vaccination. But since the COVID-19 wasn't studied that thoroughly, or we didn't have the human tests, we couldn't decide before. So we didn't have any answer. Where will it stop? From the first news, we thought that it is more contagious, and from the first deadly cases, or the media exaggerated a bit, and the mortality numbers were thought to be higher, and then everybody would think that vaccination is needed. But right now, as we can place it on this diagram, it is easy to understand that we don't need it. We don't need vaccination in this case.
Yes.
Thank you. What we have ongoing is the silent microbiome crisis. It means that the way we are meeting the nature, the contact with the nature is distant right now, and we are not seeded the natural way, which is on the left side, the normal way we are seeded with microbes. We meet them first with vaginal, the normal delivery, the breastfeeding, the breast milk is not sterile. We have to accept it. They contain all the lactobacilli, which are used to have all these oligosaccharides which are in the human milk. And they are only beneficial for the children if they are together. So the breast milk is probiotic with prebiotics. So it's called symbiotic. Symbiotic. So it's a symbiotic. It's not directly giving nutrient to the children. makes nutrient for the bacteria colonize the gut, and they are providing the essential amino acids for the child to grow. Very important thing to understand. And also one very important thing is that this propagation of bacteria, overall propagation of beneficial bacteria, is inhabiting the total surface of the epithelium of the gut of the newborn baby. And since it does this, there is no place for pathogens. They exclude the pathogens, and they are making a homogeneous, very simple but very strong microbiome for the newborn. And this microbiome can produce all the necessary nutrients for the baby.
And what about the women who don't have no breast milk or have C-section?
It is not about the woman, it is about the children. Yes, the children will be deficient, or it is an estimation also, but they say that after 12 years of life, they are getting the same maturity of microbiota as the breastfed babies. So they are more prone to allergies, to asthmatic conditions, all these, which enter through the oral cavity, because they don't have the first contact, they don't have the first colonization from the mother, and the breastfed milk is not colonizing the whole system. So therefore it is very important. The other source is the family and other animals from Gerald Darrell. I took this quotation because if we have contact with family, if we live in a countryside and we have pets at home, then it is in studies it's proven that all these children who were in close connection with animals, they are less prone to allergies, asthmatic conditions like this. Playing in the dirt, connection with nature, we have to let the children to do the discovery of the world by putting everything into the mouth. Because the mouth has this beautiful and admirable organ, the tonsil, and from the dirt he takes up the Lego, the play, puts it in the mouth and puts back all the bacteria in a very big dilution with an ecosystem, and the tonsils can take the samples and put it into the knowledge bank, and they are developing, they are educating the immune system for the specific immunity, not the non-specific ones.
Yes.
And they are taking this, but without oxygen, without all the necessary parameters, nutrients and oxygen. If we don't nourish ourselves, we are not in use of our developed immune system. So the balance should be always kept. It is never without it. And the regular ways we are taking, we have to be sure that we are taking in microbes when we breathe, when we drink, when we eat. And that is the normal way. And we have to be in connection with the microbes, because the microbes are giving us all the certain triggers I will talk about later, which are needed to have the alertness of the immune system. If you deprive yourself, like you are drinking sterilized water, or sterilized food, or sterilized air, then you are going to be like a basil you buy in the supermarket. You know these basils. You buy a plant and you are very happy that you will have the green spices at home. And you put it in your garden and they die out. Why is it? Because they are developed, nurtured in a sterile environment. And if you put it out, they don't know what to do with all these bacteria in the ground. And they die for the first fungi, or the first not really harmful enemy. That's why you cannot. If you pick it, lick it, and put it in the ground, they will have roots and they will survive. But if you put it with the roots, with this sterile ground, they will be infected and they don't survive. This is a very good phenomenon that we are taking our food and water system, we are sterilizing it, And
Everything
we are living like
Yes.
lab rats or sterile conditioning raised plants. And the last one is how we are seeded regularly is by meeting other people, the physical contact. This is also very important. What is very important is that we are making boundaries of the physical contact seeding, because we are using sanitizers, we are using antibacterial cosmetic products, and we are reducing our microbiome on the outside. And if we don't have these contact with others, and we don't have these different microbes from other people, then we are narrowing our immunity responses. Okay, next one, please. I have to come up. What I would like to show you in this slide is that we have a very, very well adapting system. In the microbiome is sampled on a daily basis, and the diversity is shown in these graphs. And what you can see is that the diversity is changing day to day. What does it mean? It means that it is adapting to the conditions we are surrounding ourselves. Mm-hmm. So if you go to a sterile environment, then it will be reduced. If you go to countryside and go into farm work or something, then it will be high. So the microbiome, the microbiota, the gut flora is reacting on the environmental stimuli in 12 hours. Mm-hmm. So they are acting very fast.
Very fast.
Very fast. Also.
Wow.
Next, please. What happens if we experience a problem like the dysbiosis? The dysbiosis is associated with a lot of medical conditions. I think we are going to share this presentation on the Kaqun website, so in order to save time, then I just ask the next slide. If we can. Yeah. What is the role of the beneficial bacteria? We call it EU biota, like EU stress and distress. Mm-hmm. Dysbiosis, eubiosis. That when they are in symbiotic relation, and towards the microbiome, the good bacteria are representing that we are here. These are the quorum sensing molecules. They are sending over that we are here, and this is our property. They are also excreting bacteriocins, which are antibiotics, briefly say, but these bacteriocins are selective. The beneficial bacteria like it. They are not, they tolerate it. Yes, but the pathogenic ones, which are not fitting into this ecosystem, don't like it. And it works other way around. When we are infected, then the pathogenic bacteria are producing bacteriocins towards the beneficial ones. Okay. So that's why we have to keep the balance. We have to keep the beneficial ones at bay. Mm-hmm. These bacteria, the beneficial ones, also produce hydrogen peroxide, which is, as you know, is very deadly to pathogens like viruses. It is very important because if you have a healthy and well-balanced microbiome in your body, and they produce all these materials like hydrogen peroxide, they are prone to get the virus infection, because they are more exposed to viruses than us. So they are the first line of defense. And they are more afraid of viruses than us. So they have their immunity against viruses. And they produce hydrogen peroxide, which kills all the viruses, regardless of the kind. Because all the bacteriophage viruses are such a big family that the bacteria, the beneficial bacteria, are providing this non-specific general target against viruses. Therefore, I wanted to show you this slide because this is very important, how the bacteria are protecting us and themselves from the viruses. And also lactic acid, they produce lactic acid. Lactic acid is very good because lactic acid controls the propagation of other microbial organisms like mushrooms or single-cell mushrooms or pathogenic others. And for us, they provide a lot of good things. They are providing us hormones and neurotransmitters. Tryptophan metabolites is a very tricky name for serotonin, which is the hormone of satisfaction. And they also produce histamine, which is very good in the respect of immunity. The histamine has a very good. If the bacteria are present, then and they produce histamine, the histamine enters into the body and makes the immunity alert. This is the bell ringing for the immunity. Why is it important? Because if the bacteria produce histamine, then you say that you will get allergies, food allergies. But if they also produce short-chain fatty acids, metabolites, which are also there, polyamines and other nutrients for the body, then they feed the epithelial cells. And the epithelial cells are producing the monoamine oxidase and the diamine oxidase. These enzymes are degrading the histamine itself. So the bacteria comes with the histamine. It shows that something from the outside world entered into the body. And if they are good ones and they produce us short-chain fatty acid or food or anything good, then the body will produce the enzyme which degrades the histamine and reduces this alertness and reaction.
Okay.
Next one. So, briefly putting together the way how the nature encompasses us is that the bacteria inhabit our internal guts. The propagation of the bacteria is always helped with the milk, the saliva, the perspiration, tears, gastric juices. All these contain materials that are feeding a good and beneficial bacteria and also the pathogens. They feed and motivate us because they give metabolites, nutrients, and also the hormones, interferons, all. And what is very helpful that they are very specific to the food we eat and very specific to the water we drink. And if they live there, they have their own metabolites. And these metabolites are tuning the digestion system, what is inside. You don't do the same digestion for meat than you do for a salad. And for what you do for an ice cream. It has to be different, the way you digest it. And what is very important, and we are doing this presentation today, is that the protection, the immunity. The immunity, the non-specific histamine ways of immunity, they are producing human defense proteins, defensin, dermicidin, all these. They are doing a vigilant and learning ability of the immune system. They keep it on, and they help the T and B cells in order to have the macrophages clean up the dirt which entered the body. Okay. The last words, we go to the therapies for this biosis, because all the therapies in the traditional Chinese medicine, they had a yellow soup which was diluted excretum, feces.
Yes.
Dried and excreted, but since the Cultural Revolution, it is forbidden right now. But it was very useful for any kind of digestive disorders. In the modern world, we have the fecal microbial transplant, which is useful in many trials. Wow, we are just the slide came back, sorry. Yeah. And we have the probiotics, which was produced by our grandmothers in fermented foods, and now we have it in capsules. And there are some fermented dietary supplements, which is also in the Kaqun's family, the Kaqun drops, which is also making the natural connection closer. We are entering again to the same way we are developed, the human body is developed.
So excellent.
That's it.
Thank you so much. So both together would be a very useful thing to do, huh?
Yeah.
Reoxygenate the cells and use the drops as rebuild the microbiome, rebuild the digestive system, very safe way. There is 60 different bacteria tribes in there, and I know you fermented them, the herbs itself, and then you have a process of colonize them and package them very nicely into liquid.
Yeah.
And that's why they are alive, is it? They are alive.
Yeah, they are alive, and they contain the process we are making it is a fermentation process, and it is also an extraction of the herbs we use. Mm-hmm. So the active ingredients are extracted and metabolized by the bacteria as well. Mm-hmm. And this is, as far as I know, this is the only water extraction of propolis, bees glue. Mm-hmm. And what I would like to tell about the bees glue is that the bees themselves use it to attach the hives on the environment. Mm-hmm. So this is the connection. So it has to be very safe and full of the material which is needed. But the bees don't have 70% alcohol in their mouth. When they eat it, when they are sick and they eat the bees glue, they don't have the alcohol, they only have saliva, which is aqueous solution. Mm-hmm. And this is an aqueous solution. It works the same way. Yes. In a lower dosage, as far as I know, the only.
Very interesting. I think we're going to need to talk about this. Yes. So if we have questions here as we see, thank you very much for listening, and thank you very much, Adam, to give us all this background on this Kaqun drops and what to lead to. So we have some questions leading there. You have a question here. What to do? Because it's going very fast.
Very fast, yeah.
Yes.
Yes.
To get vaccine for the coronavirus with things as they are present. I cannot say any recommendation right now.
No, not now, because we don't know the real numbers.
What I can do is a recommendation before they come out with a good vaccine.
Yeah.
And that is very strong. You have to sleep well. You have to move. You have to exercise. Move your body.
Eat right.
Eat right.
Yes.
Sleep right, eat right, exercise.
Nourish your body with liquid.
Nourish the body with liquid. Nutrition, yes.
Yeah.
The next one. What exactly do you mean by yellow soup? Yellow soup is a poop soup. It's made of healthy people's stool. And the Chinese doctors are collected the stool of healthy people, they dried it, and they made a powder. And if somebody had any kind of digestive issue, they put some of this powder into the soup. And that is a very traditional way.
Yes. That was the old way.
That was the old way of fecal microbial transplantation.
Yes. We have much better way right now. So.
Yeah. Can you tell us something about exosomes and their role in immunology and cell communications? Oh, well, yes, but I guess it is not we don't really have the time for that, because the exosomes and the immunology communication is another topic also, with the quorum sensing materials from the non-living parts of the environment should be noted. I guess there will be some more time later on and we can discuss it.
Here is some question. Do you think why is vitamin C doesn't work? I don't think vitamin C is not working. Vitamin C is the vitamin what is capable to actually pressing down HIF alpha 1, hypoxia factor HIF1 alpha. If that one elevates, that means you have high probability of inflammation in your body. And if that HIF alpha 1 elevates, now it's in new science, you can see that in cancer patients, they are very high. At the same time, the vitamin C is very low. No matter how much vitamin C you take, somehow the vitamin C is always low. So long as the vitamin C low and the HIF1 alpha is high, you have no chance to bring in the natural killer cells to recognize the tumoral cell or the viral infected cells. So yes, the vitamin C is good to take, but unfortunately the dosage, what they do, they usually put them into tablet form or liquid form or some kind of an intravenous form. All of them has their downwards too and some side effects. The best way to use it to put vitamin C powder like L-ascorbic acid. You can buy it in health food store. Very cheap L-ascorbic acid, and about 2 gram, 3 gram per day. You put it into coconut water, keep it there for 15 minutes, and then close it down, and you can have it for all day long. You're gonna have maximum vitamin C in your body. So please take vitamin C. It's good.
What would you advise to start with in a patient with negative test of COVID-19, but with higher level of iron, ferritin, low oxygen saturation, erythrocyte, dizziness, muscle stiffness, breathing problems?
Mm-hmm. So it definitely is not coming from COVID. So this is something else. It must be get a blood test, a full blood test, and see how the organs function, namely the kidney, the liver, see the white blood cells condition, the red blood cells, the hemoglobin, hematocrit, see all those parameters, maybe see CRP, look at the inflammatory factors, and then you can make your decision how you treat them. Yes? Next, what we have here?
What? The question and answer
yes,
are
the
populating,
UK,
not the
somebody
the
asking
chat.
about UK order. There is a Kaqun.uk. You can go on and you can purchase it there or look it up to it there. And we just want to give you the scientific background on this. What is it? But you can find it there. And every country, I don't know, many countries is joining, as I see right now from France. I see people and Canada, lots of people from Canada, and from people coming from Malaysia. My good friend, I see Dr. Saeed is a very highly respected medical doctor also, and he's listening, and maybe Dr. Nazri Mokhtar also on the list there, as I see, and the royal family I see. Somebody is there taking the time to listen to this. Thank you very much again on that. And what else? What else we have a question here? Let's see. Yes, we have a glass bottle also. We are just coming out with it because the demand is there to bring a different quality of water there. But our PET is very safe. But we know in years from now we need to change it. So we already start up to distribute the 1 liter water, and I have 1 liter and a half liter of water, Kaqun. And anything you see for yourself?
Yeah, I saw something with tonsillitis, but so many questions that I cannot track it. Oh my God.
Oh, my daughter is here, Aniko. Hello, Aniko, from Las Vegas. So people are joining up from everywhere. Excellent.
The graph says that highly contagious vaccinate, highly fatality vaccinate. Where is the cutoff? I cannot have a personal objective. I won't vaccinate our family, but I cannot say anyone what to do with vaccination. I'm not entitled to do so.
Yes.
So I'm sorry, I cannot give any advice.
Yes,
on this.
And even would be foolish to do because we don't know the real numbers.
And
so
we don't know. I don't want to be in a position with the with the last one, the swine flu. The swine flu was vaccinated in the U.S. Globally.
Yes.
And it had more side effects than good effects. And
yes.
And I don't think.
And I think it's too early to talk about that.
Yeah.
But thank you very much for the question.
And so we might just do one more question if that's okay, just because we're getting close to 2:15. So are you happy just to answer one more question from the audience?
Yeah, please highlight one, please.
Maybe what is that?
If someone gets appendicitis, it is too late to save the appendix through nutrition. Of course, it's too late. Yes, because if the appendix is ruptured, then it contaminates the whole system, and it is not the bacteria that will do the bad thing, but it will be the immune system that kills all the bacteria and makes sepsis. Mm-hmm. So the toxication.
There's somebody asking if I would take the vaccination. I'm telling you, if the numbers are matching and there's out of 100 patients, 100 and let's say 100 patients, out of 100 patients, 11, 12 patients dying, definitely would take it and support it. And otherwise, until don't reach that number, there is no need to worry about that. All you have to do is to try to keep yourself active, eat right, exercise, go to fresh air, open your window. Maybe if you have Kaqun water, maybe drink the water every day, 1.5 liters, 5 times a day, 3 deciliter. And when the time is right, you can come to one of our centers in Las Vegas or anywhere. You can come to Hungary, take the bath. Pretty soon we're gonna have it in England too. So we're gonna have able to bring you in, because they promised us this is not over yet. And please, if you have time and a chance to test it, find out about it, where you're standing. That's gonna be the key. Be tested, you know, distancing, and then you're gonna be safe. That's what it is. You have to stay safe.
Okay, great. So thank you so much, Dr. Lyons and Adam, for a very informative webinar. It's been so nice to have 2 guest speakers for our ANP webinar today, sharing real clinical insights into hypoxia, the microbiome, and immune function. What I can do is I can circulate the slides so everyone gets a copy with your CPD certificates. And we've had such a large number of practitioners on our webinar today, which is really fantastic. But if you do have questions and you haven't been able to have your individual questions answered on today's webinar today, what I'll do is I'll circulate Kaqun's email address as well, and then you can reach out to them and they'll be able to follow up with you separately. So this brings today's webinar to an end. I'd like to thank everyone for joining us for the webinar today. And I'd like to thank again Dr. Lyons and Adam from Kaqun. Thanks everyone. I hope you have a really great day.
Thank you so much.
Thank you very much. Great day for you.
Take care. Bye-bye.
Bye.